Right, so I am doing a series of videos to teach haemoglobin. So this first one is the theory of haemoglobin, and I'll be working from the PowerPoint that I'll send you as well to go through the theory. The next few PowerPoints will be working through the question pack together. So let's get started. Okay, so if you go into your student notes, you can see in your student notes that it's only a tiny bit of the specification haemoglobin, and it is in the mass transport in animals section. So we've covered circulation of blood, mass transport in animals, but what we missed out was the chemical that binds to oxygen and carries it from the gas exchange site to the respiring cell. So this is haemoglobin. So that's what we're going to start today. So some of the information that you need will be in your keynotes, and we'll flip back to them as we go through. Okay, so there's a link here to what you learnt at GCSE, something that we reviewed in our circus about surface area to volume ratio, which was just a little link to the red blood cell as a specialised cell being bioconcaved. We said this gave a large surface area to volume ratio to enable oxygen to diffuse into the cell more quickly. They also have no nucleus. So this is when they're mature. So they do start life with a nucleus, but as the cell becomes a mature red blood cell, it loses its nucleus and most of its organelles, and in that space we've got more of this really important chemical, haemoglobin, which is what this topic is all about. Okay, so we're going to focus on haemoglobin now, and I will flip between both the PowerPoint and your student notes. So the first thing, bit of what we want to understand, okay, we're going to revisit this term partial pressure, okay, today. We're going to think about the structure of haemoglobin. It is a protein, it's a very specialised protein, really useful in the essays we might be writing at the end of the course about proteins and how they've got important structure to function relationships. We've then got quite a difficult graph that we need to describe, which is called the oxygen dissociation curve, and we've got to explain why it's sigmoid shape, so why it's an S shape. So we'll concentrate on those first of all, and then we'll come back to this last set of learning objectives. So partial pressures, what did we say they meant? Okay, so we said partial pressures were basically a measure of concentration of a gas in a mixture. So oxygen partial pressure, when we're thinking about the concentration of oxygen in the blood or in the air, we use partial pressure. So oxygen partial pressure is simply a measure of the concentration of oxygen in the blood. So here's a question for you. In the blood, in this circulatory system, where would the oxygen concentration be the highest? So where is the partial pressure of oxygen the highest? Which site? I'm hoping you'll have guessed at the gas exchange site, where oxygen is diffusing from the air in the alveoli to the blood. This is where the partial pressure of oxygen is at its highest. Where would it be at its lowest? I'm hoping you're thinking at the tissues, where the cells are respiring, they're using the oxygen up in respiration and keeping the concentration really low. So that means the partial pressure of oxygen is very low at that site. This is really important to understanding haemoglobin's function. It's a very unusual protein because in these two conditions it behaves in two different ways. So where the partial pressure of oxygen is really high at the lungs, haemoglobin binds to oxygen more readily. Whereas where the partial pressure of oxygen is lower at the respiring tissues, it binds less readily to oxygen. So to understand that, we need to understand a little bit more about the structure. Okay, so there are lots of different haemoglobins. Okay, so different organisms have haemoglobin. So obviously humans have haemoglobin, but worms have haemoglobin, llamas have haemoglobin. Okay, so it's many different different organisms that have haemoglobin, and it does the same job of binding to, transporting and releasing oxygen to the respiring cells. But what kind of molecules? What did I say it was? What kind of molecule? Group of molecules. And how would they differ from each other? Because we know what kind of molecules they are. Okay. And it's all mammals and almost all vertebrates that have got haemoglobin. Okay, so thinking about all mammals and almost all vertebrates as well. So anything with a backbone basically has haemoglobin. So you've had a little bit of time to think about those first two. So what kind of molecules did we say haemoglobins are? Well, they're proteins, and proteins that are slightly different from each other in each of these different organisms. So human haemoglobin is a little bit different to the haemoglobin you find in the groundhog, and that's a little bit different to the haemoglobin you find in a whale. So knowing what you know about proteins, how do you think they differ from each other? That's right. I'm hoping you're thinking by their primary structure being a little bit different. Okay, so each one of these species will have a slightly different primary structure to the polypeptides that make up its haemoglobins, giving it a slightly different overall structure, and that might affect how they behave a little bit. So in other words, those molecules might be a little bit better for the gas exchange system of that particular species and the habitat that it lives in. But we'll come back to that idea later on. So if we go back to our learning objectives, okay, we know what partial pressure is now. So partial pressure is concentration of a gas in a mixture. So we're thinking about oxygen, partial pressure of oxygen in this topic, and we know that where the site of gas exchange is, that's where it would be highest. So that's where in the lungs tissue for a human, the blood in the lungs, but also that it would be at its lowest where gases are being used at the respiring tissues. So we need to look at this structure of haemoglobin now. We know that different organisms have different shapes of haemoglobin. We know they have different primary structures, but we need to look at the overall structure. So a very common question on our exam is when the examiner says to you, haemoglobins are proteins, so they're found in all these different organisms. They have the same job, they transport oxygen from the gas exchange site to the respiring tissues, and they all have quaternary structure. What does that mean? Well, we know it must mean that they're made up from more than one polypeptide chain, because that's the definition of quaternary structure. So if we look at that structure, because this is haemoglobin here, it's human haemoglobin, how many polypeptides do you think make it up? They're labelled. So you should count 4 in total, 2 beta and 2 alpha. And each one of them has got its own heme group. And a heme group contains iron. So it's an iron containing group. And it's that part that associates or binds to an oxygen molecule. So each haemoglobin can bind to 4 oxygen molecules. So each heme group can carry 1 oxygen molecule each. Okay, so what's the role of haemoglobin? I've told you many times in this video so far. To carry oxygen from the gas exchange site to the tissues that need it for respiration. How many oxygen molecules can a haemoglobin carry? Related to the heme group, so how many was that? 4. A single oxygen molecule combines with a heme group. So 4 oxygen molecules can be carried by 1 haemoglobin. Even Charlotte got that one right, well done. Okay, when haemoglobin combines with oxygen, no matter how many oxygens, we stop calling it haemoglobin and we call it oxyhemoglobin. So sometimes you'll see that term in exam questions, and it just means oxygen is being carried by those haemoglobin molecules. Now, this is always the thing that I think you should really stop and ponder about, because this is an amazing protein. Okay, not only has it got quaternary structure, not only has it got additional groups that were added to those polypeptide chains, those heme groups containing iron, it has to do something really clever. To carry oxygen from the gas exchange site to the tissues for respiration, haemoglobin must load oxygen really readily at the gas exchange site, so it must bind to it really quickly, but it must do the complete opposite at the respiring tissue sites. So it must unload oxygen really readily, so what we say, disassociate from oxygen, unbind the oxygen really quickly. Okay. Why can you do both? That's because it's a protein. Okay, so as I said, they're amazing. So we've already talked about partial pressure of oxygen. Okay, so this next bit is a really important graph. So what I would like you to do now is just pause the video, and I want you to make a sketch of this graph on your paper, including the axes. Okay, so now you should have joined me again, and I want you to do some identification on your graph. Okay, so first of all, let's read what the graph says. It says that at the bottom on the x-axis we've got partial pressure of oxygen, which we said was the concentration of oxygen, and you can see that's increasing, and you can see that as it's increasing, the other thing that we are measuring is the percentage saturation of haemoglobin. So saturation is a way of saying how much oxygen it carries. So the higher saturation it has, the more oxygen it's carrying. So the most saturation that could be possible would be 100%. And that would mean every single haemoglobin molecule in the organism's blood was carrying 4 oxygens, fully saturated. Now, in reality, that never happens. This is about 98%. You only get to about that kind of saturation. Okay, so on your graph then, I want you to have a look at the saturation, and I want you to think about where it's highest, and then I want you to have a look at partial pressure, where that's highest, and is there a relationship between those? Again, look at partial pressure when it's lowest, and have a look at saturation. So where did we say partial pressure was the highest in the circulatory system? In the lungs, yes, that was it. And where did we say, or where does the graph tell us, sorry, that saturation then must be highest? So if we read on the graph, partial pressure is the highest there. Where's saturation the highest? Well, that is also when partial pressure is at the highest. Okay, when's partial pressure the lowest in the circulatory system? In your tissues, yeah, and that equates to where haemoglobin saturation is the lowest. So the graph confirms what we said about haemoglobin and its special properties earlier. It's telling you that when we are at the tissues, the haemoglobin has a really low saturation for oxygen. So in other words, most of the haemoglobin is letting go of its oxygen. It's disassociating, or unloading is the other thing that you can say. So it's unloaded its oxygen, that's why its saturation is low. When the partial pressure is high, so like in the lungs at the gas exchange site, then the saturation is high. So that's confirming that in those conditions where there's a high concentration of oxygen, the haemoglobin is binding really readily to it. That's why its saturation is so high. Okay, so a really good word to use to help us explain that relationship is the word affinity. And affinity is a chemistry word, really, and and it means that two things are attracted to each other. Okay, so affinity, not like Buzz Lightyear, Affinity and Beyond, no, but well remembered. Affinity is like an attraction, so I've got a high affinity for Charlotte. Who is videoing. So thinking about haemoglobin then, why is the saturation so low when the partial pressure is low? Well, in low oxygen conditions, haemoglobin has got a low affinity for oxygen, so it doesn't bind to it very readily. It's more likely to unbind and unload the oxygen. But as you increase the partial pressure of oxygen and the oxygen concentration becomes higher, that changes the property of haemoglobin, and it becomes has a higher affinity, binds to oxygen more readily. So oxygen is rapidly associated with haemoglobin when the oxygen levels are high, so in the gas exchange sites. And oxygen rapidly dissociates, from haemoglobin when oxygen levels are low in the respiring tissues. So that's the rules, that's the way haemoglobin behaves. But it's not a straight line. So that relationship is not a linear relationship. That curve is in F shape, can you see? So it's called a sigmoid curve. Okay, and you've got to be able to tell the examiner why it is that shape. So it's not going to be good enough just to say at low concentrations of oxygen, haemoglobin's got a low affinity for oxygen, therefore a low saturation, and at a high partial pressure for oxygen, it's got a high affinity for oxygen and so a high saturation. That's not going to be enough on its own to explain the shape of that graph. That would be a linear, I could describe a linear relationship. So you've really got to think about protein structure here. Okay. So if you think about the first oxygen binding to the first heme group, I want you to imagine that when that happens, the structure of the haemoglobin just changes a little bit. Now, of course, we know that when molecules bind to proteins, they do change shape. We know that from our enzyme-induced fit model. So imagine the same sort of thing. An oxygen, the first oxygen, this might hurt actually. Can I put it to anything? No, that would hurt. What can I use as oxygen? Yeah, okay. So first oxygen binds to the haemoglobin, and it's going to change the shape of the haemoglobin, so the binding site for the second oxygen becomes more prominent and is more able to bind to the second oxygen. Okay. So the first oxygen binds the haemoglobin, it causes the shape to change, allowing more oxygen to bind more easily. Okay, so you get a greater saturation with oxygen. The name of this is called cooperative binding. So the binding of one oxygen increases the chance of the next one binding. So if you think about it, as it becomes saturated, then you can see it's more likely to bind oxygen, and that's why the curve's becoming steeper and steeper until you get to the last binding site. Because you've got 4 binding sites, if 3 of them are occupied, the chances of oxygen colliding with and binding with that last one are a bit lower, so it starts to then level off. Okay, so the relationship shown in this graph is called the oxygen dissociation curve, and we really need to understand that graph structure. Okay, so could I ask you to have a go at, we'll do it in class on whiteboards, but I'd like you to do all of those answers on paper, and we'll check in with me in a minute, okay? And you can check the answers. Thank you. Okay, so partial pressure, what did that mean? Okay, so you should have in your answer that this is a measure of the concentration of a gas in a mixture, and if particularly you're thinking about oxygen, it's the concentration of oxygen in the blood. What's the role of haemoglobin? We said it was an unusual role because it had to bind to oxygen at the gas exchange site, then carry the oxygen to the respiring tissue where it unloads the oxygen there to be used by the cells in respiration. Okay, so sort of three things to think about in that. It's got to bind to oxygen at the gas exchange site, transport the oxygen to the respiring tissue, and then unload the oxygen to the respiring tissue so it can use the oxygen in respiration. What types of organisms have haemoglobin? You should have all mammals and most vertebrates. What type of biological molecule is haemoglobin? Protein, and if we describe its structure, it's a protein with quaternary structure made up of 4 polypeptide chains, 2 beta, 2 alpha, and each one has a heme group containing ion. Each ion containing heme group can carry 1 oxygen molecule. So each haemoglobin can carry up to 4 if it's fully saturated. Okay, right, so we're going to sketch our oxygen dissociation graph now. So on the x-axis, partial pressure of oxygen. On the y-axis, percentage saturation. Our S-shaped curve, obviously coming in below. If this was around 98%, that's when it would start to level off. Okay, so what we'd be showing is that if we go for a low partial pressure of oxygen, we've got a low saturation, and the reason for that is that under those conditions haemoglobin has a low affinity for oxygen. Okay, the opposite condition, here we've got a high partial pressure of oxygen and it gives us a high saturation. Okay, so haemoglobin under those conditions is said to has a high affinity for oxygen. So we can link these conditions to a property of the haemoglobin which gives us an observable, sort of measurable change in saturation. So in areas like respiring tissues which have got a low concentration of oxygen because the oxygen is being used up in respiration, under those conditions haemoglobin has a low affinity for oxygen and so unloads the oxygen to the respiring tissue. Under the opposite conditions of high partial pressure, the haemoglobin has a high affinity for oxygen, so it will load it readily, and that explains why it's nice and fully saturated nearly under those conditions. But then we have to explain the S shape of the graph. Okay, and the S shape, the sigmoid shape, can only be explained when you start thinking about how that affinity changes. So as the first oxygen binds to haemoglobin, you get a change in its shape. Okay, and when it changes its shape, what happens is the second binding site becomes easier for oxygen to bind to. So that says making the second binding site. Easier to bind to. So the haemoglobin affinity for oxygen has increased. Okay, so it's increased. Right, we'll pause there. Okay, I've just pulled up this animation to be able to show you this change in shape, or what is referred to as a conformational shape change in the haemoglobin when oxygen binds to it. Okay, so you should recognise that we've got our quaternary structure, got our 2 alpha and our 2 beta chains, we've got our heme groups here. Okay, so just play and pause. So in the animation it shows oxygen binds into the heme group. So there's a heme group, there's the iron atom, and an oxygen molecule can bind to that. That will be the first oxygen that binds. Oxygen, when haemoglobin's got no oxygen, we can call it deoxyhemoglobin. Okay, so in the lungs we've got a high concentration of oxygen. So just pausing that there, when that first oxygen bound, it caused a shape change in the structure of the haemoglobin, making it easier for this second binding site. Okay, we call this a conformational shape change, making it easier for the oxygen to then bind to the other heme groups. Okay, you can see that happens for our second and third binding sites. But then when we get to that fourth one, can you see that got a little bit harder for that final oxygen to bind to, just because there's fewer binding sites available. Okay. And now we've got a fully saturated oxyhemoglobin molecule. So you think about the oxygen dissociation curve then and its shape. That conformational change is the explanation for it, okay, which we call cooperative binding. So first molecule joins quite slowly, second and third faster, fourth more difficult again, giving us that S shape. Now that's quite a difficult concept, okay, and I'd imagine doing it like this where you can't ask questions might make it seem quite a lot trickier. Okay, so let's just have a little look back through the keynotes and just do a summary. Okay, so in your keynotes it does build up a little bit slower, okay, so you can read through these as well. So we've got information about the shape of haemoglobin, it being a quaternary structure, knowing that this molecule is has heme groups and that each heme group binds to one oxygen. As we've said, oxyhemoglobin is formed, so oxygen joins the hemoglobin when the hemoglobin associates. You can use the word associates or you can use the word loads oxygen, okay, and it does that in an area of high partial pressure of oxygen, like the lungs, the gills, any gas exchange sites. Oxyhemoglobin disassociates or unloads oxygen in areas of low partial pressure, which are the respiring tissues. So then we summarise that in the graph for you. Okay, so at high partial pressures of oxygen, haemoglobin has a high affinity for oxygen, and so it loads it more readily, so it binds to it more readily. And we know that because at this point it's got an affinity, a shape that means that it can bind more readily, giving a high saturation at those high partial pressures. At low partial pressure of oxygen, haemoglobin has a low affinity for oxygen and unloads more readily, giving a lower percentage saturation of haemoglobin for example, respiring tissues. Why is it S-shaped? This is the really quite difficult concept, and it might take you till year 2 to completely get your head around this. And I wouldn't be surprised if there's some second years watching this thinking, I wouldn't mind recapping haemoglobin in full now, because this bit, the first time they met it, would have been quite tricky. I imagine now they're thinking, oh, no, makes sense with everything I know about proteins. So, why this shape? So we said, the first molecule of oxygen, it was quite hard for it to bind, and that's because those subunits were all packed closely together. But the structure changed when the first oxygen bound, and that makes more oxygen binding to the molecule easier, so it gets more saturated. So the second and third one will bind more easily, and so that's why this middle bit of the graph is steeper. And we call that cooperative binding. So the binding of one molecule makes it more likely that another and another will bind. And that continues really until you're almost fully saturated. But then it does get a little bit harder for the fourth molecule to bind, just because there are less free binding sites. And that accounts for it just slowing down a little bit there in the curve at the top. Key terms there in the box, so you know what you're using. You can either use loading or associating. You can use unloading or disassociating. They mean the same thing. And then affinity is attraction for something. So we're saying if it's got a high affinity, it's going to bind more readily to it. Now, so far, they have actually asked students to sketch an oxygen dissociation curve in the exam, and they did this last year, and very few students got it correct, because quite a lot of them took their line to all the way up to 100 when it should be below 100, and not many of them did this wonderful S shape, which we've spoken about today, or I've spoken about today. So it's making sure that it's always an S shape and that it does not go all the way to 100%. Okay, right, so we're going to stop this video now, and that'll be our first lesson.